Good morning, everyone. So we'll just get started with the WRI webinar. And presenting today is Melanie Ford, and she's a third-year PhD student, almost finished, and is presenting her research work prior to thesis submission in December. And she'll be talking about plasma treatment of post-harvest grains. Just hand it over to you straight away. Thank you. Share the screen and
all right, so getting started here, I should probably give a little bit of background. If anyone has seen some of my presentations on campus in the past, you're probably wondering why in the world I'm talking about post-harvest grains and plasma treatment when all of my previous research has been on root exudates. Well, to answer that question, it has to do with COVID. Last year, a lot of my interstate and international collaborations for the PhD, which were absolutely essential, fell through, and I had to rapidly change gears. So did a bit of brainstorming, worked with my supervisors, and we decided to take a side project and turn it into the main thing, which means we've had to do a lot of development, and I've had to learn a completely whole new field of research. And in order to do that, we had to bring in a lot of different groups. So not only am I a PhD student at the University of Adelaide, I'm also a PhD student with CSIRO Health and Biosecurity based out of Kintore Avenue, and I'm a visiting scientist with SARDI working with the serial pathology group. Like I said, we've had to kind of put together this massive team, and I've also had to gather the campus and kind of get everyone to rally to help me get this done in such a short time period. So this presentation is just to kind of share with everyone all of the work that we've done together. And so everyone knows all those bits and pieces, they can kind of see what's happened with them. Now, before I can get started here, I should probably let everyone know that this work has not been published yet. Most of this data has been collected within the last 14 months. And so we just ask that no one shares it, not just yet. If you do have any questions or comments about sharing it, feel free to contact me. Now, the world's got a lot of problems right now. There's a few big ones that are looming over us that are always in the media, and there's some that have kind of lost traction over the past few years. And one of those is food security, which is a massive issue globally. So our food crops, predominantly cereals that are the staples of people's diet, including wheat, maize, rice, and other grains. Well, over 20% of the yield of cereals every year globally is lost due to fungal pathogens. That is an incredible amount of food, causing billions of dollars of economic loss every single year. And with over 800 million people going hungry per year, this is a big problem. Now, we have lots of ways to work around this and try to mitigate some of these pathogens. A lot of them involve the use of fungicides and things like that, proper storage practices, hygiene within storage and transport. But that doesn't always work. In fact, there's a lot of fungicide resistance. There's also a lot of new regulations evolving around residues left on the grains from some of the sprays that we're using. There's the cost of it, and there's all kinds of storage issues in developing countries. But again, it's a really big problem, so much so that in 2015, the UN Assembly set out 17 sustainable goals for 2030, and 2 of them, according to the Food and Agriculture Organization of the UN, 2 of them are related to microbial infection of grains and crop loss due to those. So goal number 2, which is zero hunger, has to do with a lot of that. Same with goal number 12, which is responsible consumption and production of food. So there's still a lot of work being done. And as we switched gears with my project, my team of supervisors and I kind of sat down and thought, oh, well, is there a way we can start to address some of these things in a big multidisciplinary collaborative project? And so we came up with a few questions. And one of them is, can we get rid of some of those fungal pathogens on the grain? Can we knock them back or just completely get rid of them? Is it possible? If so, can we do it prior to storing the grain? And can we do it without any chemical input or output? So we thought about it for a bit, and we thought, well, there's plasma technology. plasma might actually work. Now, the word plasma is being thrown around a lot in the media lately, and we're not talking about the liquid part of your blood. That is a completely different type of plasma. What we're talking about today is the forced state of matter. So this is all physics. This is a bunch of electrons and ions bouncing around all over the place. And really what that means is that there are 4 different states of matter that we know of. The first is solid, if you think of an ice cube. The next is liquid. Think of water. If you continue to energize that, it turns into a gas or water vapor. And then if you continue to energize that water vapor, that gas even further, it begins to split all of the molecules and all of the atoms even apart, completely ionizing it. And that's what plasma is. In fact, as you can see from the illustration here, it's often found in neon signs. But that's not the only place you'll see it. In fact, it's super common in nature. So every time you see the sun, that is a big giant ball of plasma in the sky. Same with the stars, aurora borealis, and of course, lightning. This is all plasma. But outdoors isn't the only time you'll see it. You'll see it indoors. In fact, a lot of you are probably in rooms with fluorescent lighting directly above you. That itself is plasma. Again, those neon signs, old school TVs from the early noughties. And there's a really cool thing that's being used very often at the moment, and that is plasma in a benchtop kind of microwave looking thing. And it's used for the sterilization of medical equipment. So you can put surgical instruments and things like that in there, turn it on, zap them with plasma, and it gets rid of the microbes. So we thought, well, could we do that, but do it with grain and keep the grain moving so it's always turning and moving through the plasma instead of putting it into a box? And what we did was start to use kind of this Frankenstein version of a rotary evaporator. So what it is this big rotary evaporator. It's got some electrodes strapped onto it. You have a flask full of seeds. It was originally developed in 2015 by a different research group based at UniSA. We've made a few modifications, chucked some grain inside, and voila. But how is that actually working? How are those field peas in that middle of that flask that you can see just like, what is it doing to them? Well, we know that within plasma, there we go. Within plasma, there's a lot going on. It's essentially this massive, almost invisible tornado of tiny, tiny little reactions going on. So all of those ions that are bouncing around in electrons, every time they start to hit each other and hit those seeds that are inside, they physically start to bombard things, creating all kinds of like physical etching. We also have chemical reactions that are happening. So all those little bit of fragments that are being etched off of a surface, like the grain, they start to then become chemically reactive. And because we're ionizing air, it means we have a lot of nitrogen and a lot of oxygen. And with all of these reactions, we're creating reactive nitrogen, reactive oxygen species, and we're oxidizing the crap out of all of the things we're breaking up on the grain surface. There we go. So if we're going back to that rig we're looking at, inside this flask with this grain spinning around, we have all of those reactive species. We have ions, we have electrons, we have neutral fragments in this giant tornado of lightning going on in this jar. And we think that this might be enough to actually get rid of the microbes. And the best part about it is that we're only using the air in the atmosphere around us. And as soon as you turn it off, all of those reactions stop and we're just left with the grain. So is it a solution? Is it going to work? Well, we're not really sure. Plasma is used, again, to sterilize medical equipment, and it has been used on grain a lot in the past. There are plenty of papers on this, some dating back all the way to 1961, when people were chucking rice into plasma generators. But a lot of the work that's been done, some of it has been to reduce the microbial load, but a lot of it's working on increasing germination rates. Now, the biggest problem with all of that is the research is completely full of gaps and holes. It's like Swiss cheese. There are massive variations in all of the different plasma equipment people are using. They're testing random species and not really going into depth of the biology of those species. And they're making some huge claims, like plasma is a cure-all, it's the best thing for germination. So we really wanted to look and kind of think, is it really true? So for this PhD, we decided to do a systematic study. And really, we're trying to link all those physical and chemical changes made from the ionization in plasma back to what's happening on the surface of the seed. We're also trying to understand how that impacts seed viability, growth, and development. And then the most important part, in my opinion, is trying to use that plasma to get rid of grain pathogens, all of those fungi off the seed surface. Now, before we can really begin, I should probably lay out our usual suspects. These are the model species that we chose for research. And a lot of times people would choose 1 or 2. We decided to go big and go with 5 because we wanted to look to see how plasma is interacting with multiple different grain types and multiple different surface chemistries. So starting out, we have maize, which is a pretty big, robust cereal grain. It's grown all over the world and it has a nice thick outer waxy layer. Then we have wheat, which for South Australia is quite an important crop. Again, grown all over the world, but it's got a much thinner waxy layer protecting it, and the embryo is actually perched right on the surface. And that's because there are thousands of years of domestication. We've bred to have different structures on the wheat grain. Barley is similar to wheat, but it's got, well, this cultivar at least has a nice protective outer husk around the outside, so that might be shielding it from some of the ionization reactions. Then we have kind of our oddball, which is Psyllium. And if you're not familiar with this grain, it's what they make Metamucil from. So it's this little seed that when you put it with water, it basically expands into this super goopy gel mucilage. And this mucilage itself is incredibly important. That's really what the grain is used for. But it also means that the entire surface of the seed is coated in sugars instead of waxes. And finally, we have our token legume, which we're using, which is field pea, which is a nice big seed coat. Now, the general structure, and forgive this very rudimentary diagram, but we have all grains and all seeds really contain 3 main parts. There's usually an outer protective layer around the outside, often a seed coat. You generally have an endosperm or food for a baby plant. And then finally, you have the embryo or germ. That's where the baby plant is. And some of these different parts of the grain are pretty important. So I should highlight where the locations are and what all of those mean. So on this diagram, I've pointed out where the embryo is. And this is going to be important with how the plasma is reacting with the surface of the grain and potentially biology. So maize, if you look at the cross sections I've made down at the bottom, maize you can see has a big giant embryo and there's some nice layers that are protecting it. Wheat, that embryo again is perched right on the surface. There's not a lot of protection. Barley is similar to wheat, but again, it's got that husk. Psyllium has almost a little smiley face looking embryo right in the middle, and it's protected by these armored layers of those sugars around the outside, plus a seed coat and other parts of the grain. And then finally, we have field pea, which you can see that nice, beautiful, thick seed coat on. So using these, we wanted to see how the plasma impacted some of the properties of seed. And the first thing to do was actually start to just scan it under a scanning electron microscope. We wanted to see what really was going on here. So it turned out that when you scan a wheat grain, this is the embryo part of the wheat grain. When you scan that, the untreated ones look nice and healthy and happy, and plasma treated ones look awful. So you can see that embryo is horribly damaged. Not only is it smashed up, but it's lost part of the outer layers of the pericarp protecting the embryo. That didn't look good. Then we decided to go on to the other side of the grain, which has the structure called the brush. So wheat grains have that hairy bit at the end. And on untreated, you can see it beautifully. But after plasma treatment, we have shaved it all off. So plasma is doing some serious damage to the outside of this grain. Looking at field pea, that was really astounding. On field pea, you can see that there's these nice star-shaped cells, the macrosclerid cells on the upper surface of the field pea. These are coated in a wax. When you put them through another series of treatments, which is tumbler and vacuum. So that means I put them into the spinning and vacuum mechanism of the plasma tumbler without turning on the plasma. The tumbling mechanism does break things apart, and that might be responsible for some of the damage we just saw in the wheat. But then when you turn on the plasma, it completely gets rid of all of that waxy surface. So it's just these like skeleton remnants of those cells left behind. And that started to make us question what was going on with germination. Would all of that damage to the grain actually impact the health and viability? So I did germination assays on the 5 different species, and we had incredibly mixed results. So maize was kind of a weird one. Starting at 15 minutes of plasma treatment, we had incredible germination rates. We were all the way up to 90%, which was beautiful, but our untreated was way, way down. And the explanation for that was, it turned out the lot of maize grain we were using was horribly infected with what appeared to be Rhizopus oryzus. And so when we were trying to germinate, the untreated grains had so many microbes on them that they couldn't take off. The microbes were getting to them before the grain could germinate. With wheat, it's a very different story. We noticed that we had ideal spot-on germination in all of our controls, and then as you start to increase the time point of plasma exposure, it drops down the germination rate significantly. 64% off a 90-minute treatment is not so good. But it also does show that a short time period of plasma, potentially 15 minutes, might be ideal. Barley, across the board, the differences were insignificant, but there is certainly a drop from 89 and 90% all the way down on the 60 and 90-minute plasma treatments into the 70s. Now, it doesn't seem like a lot, and it's statistically insignificant, but I'm sure brewers would not be pleased with those kind of numbers. Field P was a bit confusing. We're not quite sure why 60 minutes dropped down germination, but it was back up at 90 minutes. And Psyllium did not appear in any way to be affected by this. So we also looked at seedling growth, kind of measuring the plants over time as they started to grow. And with maize, as you can see, the untreated and the short time periods of plasma treatment, well, 30 minutes, those plants took off and they grew really well. But the longer plasma exposures were a little bit stunted. Wheat displayed the same type of phenomenon. Plantago, there was absolutely no difference, and Field P was a little bit crazy. I could not get a single one of the Field P plasma treated grain or seedlings to actually grow past germination. So once the first true leaves erupted from the seed, they stopped and they developmentally did not change at all, which really left us scratching our heads. So I started thinking, okay, well, we have a lot of damage to the embryos in some of these seeds. Is that going to be responsible for some of the weird germination and growth that we're seeing? And so what we did was extract the DNA from a bunch of germinated seedlings and did a deoxyguanosine assay, which is binding to all of the oxidative fragments of DNA. And it turns out that doesn't really seem to be the case. We're not quite sure. This data set's incredibly convoluted, and we can't really get a lot of meaning from any of it. Then we started thinking, okay, well, maybe it has to do with differences in hormones. The plasma might be oxidizing things. So if we're oxidizing them, the plant might be responding in a different way. Turns out ethylene production is a good response to oxidative stress in seedlings. So we decided to do a bunch of different treatments with both wheat and maize and do ethylene as well as an ethylene inhibitor to see if there were differences. And across the board, again, inconclusive. We're not really sure. So what does that all mean? Well, we know that plasma treatment has reduced germination rates, especially in wheat. Is it related to damage to the embryo? Or is it possibly related to physical aspects? Are we changing the direction and the way that water is entering the grain? Also, with maize, it had delayed early growth, but I did a full pot trial where I grew maize from seed to seed. And after about a month or so, the plasma-treated plants actually catch up to the rest of their counterparts that are untreated. And so in the long run, by harvest, there's absolutely no difference between the plants. We also know that growth and development is a bit reduced. We weren't sure about what was going on with signaling as well as oxidative stress. And so we decided the next thing we need to do is really dive into some of the chemistry, what's actually going on within the plasma. And we know that what's happening is there's all the ion bombardment to the surface. So one of the first steps I took was actually popping all of these seeds into a confocal profilometer. And what that means is I popped them into this instrument that shot lasers at the surface to see how the surface was changing. Was that ion bombardment really messing it up? And it turns out, yes. So untreated grains have this nice undulated surface. And then after plasma treatment, it turns mountainous and crazy. The topography completely changes. If we compare that a little bit further here, so if we're looking at untreated tumbler, vacuum, and plasma and comparing it to scanning electron microscopy, we can really start to untangle the differences. Untreated, again, have those nice undulating surfaces. Tumbler and vacuum, you can see there's mechanical damage happening, but plasma looks very, very different yet. So not only are we getting mechanical differences, but we're getting a level of etching. And that made us think, we need to look at chemistry. If we are etching and chemically oxidizing the surface, we need to see if there's a difference. So we used an instrument called TOFSIMS, which is time of flight secondary ion mass spectrometry. And what that means is we're basically taking a bunch of ions, we're bashing them against the seed surfaces, creating fragments, kind of a little bit like the plasma treatment, creating fragments, and then capturing those fragments in a column. And we're looking at the differences in mass of those fragments. And so we can then compare those fragments between all of the treatments and all of the species to see what's happening. So the principal component analysis on the left-hand side, that basically is just telling us that the dark blue, so the untreated, are completely separating themselves from the plasma treatment. There are really big differences in the surface chemistry between plasma and untreated. Well, that's a good sign. If you look at the heat map over on the right, it's a little bit confusing, but what I've done is highlight some of the different, I guess, fragments coming off here. So these are just more hydrocarbon fragments that we're looking at in this section. And you can tell that on plasma treatment, we've really reduced some of those fragments that are coming off. They're really high and untreated across all species, but not so high on plasma treatment. And to further complicate matters, we started seeing really weird signaling with some of the potassium. Across all of the grain types, there were massive increases in potassium rates visible on the surface. So again, that really left us scratching our heads. But it also made us think that, okay, if we're changing the surface chemistry so much, how is that changing the hydrophobic properties of the grain surface? Grains generally are covered in, apart from psyllium, the ones that we're working with are covered in these outer layers of waxes. If we're chemically altering that, what does it mean? So we decided to do a contact angle measurement. And Mike McLaughlin's group was so lovely. They let me into their lab to use a bunch of their different equipment. The first one here is this tensiometer. And you can see in the video, what I did was put water droplets on top of grains and watch them absorb into the surface, but also calculated the angle that's contacting between the water and the seed. And as you can see, untreated has this nice, beautiful bead of water perched on the surface, whereas plasma-treated water just soaks right in. And as it begins to soak into the grain, that again made us very curious. Okay, if the water's going in quickly, does that mean it's completely changing the rate and the capacity of water uptake of the seed? So the next step was to do a water sorption assay. And again, with Mike McLaughlin's group, they have this awesome new sorption meter. And what we did was put all of these grains into this thing. We sent it around at a controlled relative humidity where you can set the number of hours you want to measure, and it takes weights every 10 minutes and images every 10 minutes for that entire time under a controlled environment. So we were really able to get into detail and capture not only the rate of water uptake, but the capacity of water uptake and how that changes with all of our treatments. So as you can see, for both wheat and maize and really all of the species, untreated are that navy blue line down at the bottom. So that untreated grains have a slower rate of water uptake and lower capacity, whereas we increase with increasing times of plasma treatment. And that's really, really visible within the 90 minutes, which is that big gray line at the top. So that left us thinking, okay, if we're changing the way that water's being brought into the grain, well, does that mean we're changing the direction that water's entering the grain as well? Which is a pretty big deal. So all of these grains have over time evolved incredibly clever ways to soak in water. In fact, they have a lot of different directions they should be soaking in water in order to do the things that they're meant to be doing. And they generally want to hydrate the embryo part first, and then have the food start to hydrate, the endosperm hydrate second. Well, what we did to kind of see what might be happening is put these grains into a high molecular weight dye in an aqueous solution and see how the grains were uptaking water or water and dye. But really what this is telling us is if there's massive damage to the grain and changing the direction of water uptake. So this dye, like I said, is a really high molecular weight. We're talking about 960 daltons or so. So it's absolutely massive, which means it's only going to be traveling into areas of the grain with a lot of damage. And as you can see, for both wheat and maize, untreated and tumbler and vacuum grains after 24 hours really didn't take a whole lot of dye up. The wheat did in the brush, but that area has a lot of damage to begin with from all of thresh. So what that happens during threshing. Then when you come to plasma treatment, it's completely different. So 30 minutes of plasma on maize didn't change the direction of water uptake. That still happened in the normal fashion, but you can see the grains really started to turn blue. By 90 minutes, the whole grain was blue. And the same with wheat, but we did it in an incredibly fast manner. In fact, 30-minute treated wheat was completely blue after an hour. 90-minute grains completely absorbed the dye and turned blue within 5 seconds of adding them to the dye. So that tells us, again, we've really changed something on the surface. And we wanted to investigate this a little bit further with psyllium, because remember it has that sugary layer on the outside? Well, it turns out that psyllium grains have this mucilage, and the outer layer tends to be pectinaceous and somewhat soluble, whereas the inner layer is made of a lot more heteroxylin and insoluble. So when you hydrate it, as you can see on the untreated, it should make this nice gooey blob around itself. Then when you treat it with plasma, it seems that we're actually completely removing some of that outer pectinaceous layer. And by 90 minutes of plasma treatment, there's nothing really soluble left. It's just this dense blob of goo surrounding the seed. So I did some mucilage water sorption capacity assays, and it turns out, yes, plasma has changed the surface so much that we've completely reduced the mucilage water absorption capacity, which may have impacts on how this grain is used, since it's grown specifically for its mucilage. And finally, some of the information we were looking at, the damage to the grain that we could see on the SEM, as well as those dye assays, we knew that the grain was being damaged so much so that these gigantic molecules could enter. But that also meant that molecules inside of the seed could leave. In fact, we started to see this trend when you plate grain out onto a substrate and leave it there for 24 hours. Afterwards, it's leached, plasma-treated seeds have leached all of this dark and kind of browny yellow substance out onto the media. That didn't look so good. So we assumed there were plenty of solutes, potentially all the intracellular contents maybe leaking out. And we thought that could be a lot of osmolytes and potentially also potassium. Going back to the top sims, remember we saw those massive spikes in potassium. So to see what might be happening, we did a quick flame photometry assay where we soaked the grain overnight and then took the extracts, the water the grain was soaking in, and then tested them with flame photometry to analyze them and see if we were getting increases in potassium. Now this graph is slightly convoluted, but what it does show is that across the board with all of the species we looked at, we have increased the amount of potassium that's leaching out of the seed with plasma treatment. So that's a bit of a red flag for us. And in general, what we were able to figure out is, yes, we know ion bombardment is physically etching away the outermost surface of the grain. By removing this layer, we're changing the way that water interacts with it. And that's both the way water is moving in and around the grain, as well as the rate that it's going in. We know that we're getting things that are potentially leaching out as well. And that may actually start to explain why we're having differences in the germination rate. But we also wondered, if we're changing the surface of the grain this much, how is that going to impact the teeny tiny microbes on the surface? So this is the fun part, the part we've been waiting for, all of the microbiology, but we're going to talk for the rest of this presentation all about disinfesting grains from the fungal pathogens. Now, before we can get into that, I do need to explain a little bit of microbiology terminology, and that is colony forming units per milliliter. Really what that is a way to measure and get the concentration of the number of viable spores or hyphae. So the viable beats of a fungus that are left, the number of those that are on a grain, remaining on a grain after treatment that can grow into a new fungus. So that's really all we're talking about. Now, to get started, what I did was take all of our species. I started then extracting all of the microbes on the surface and trying to regrow them and see how many colony forming units I could get. Now, this was kind of a pilot trial, just seeing what was going on with plasma treatment. And it turns out that for each species, it's really, really different. It's not super surprising considering each species might have different pathogens and each species also has a very different surface chemistry, but it was still a little bit weird. So with maize, you can see we're really dropping things down. With wheat, there's actually a statistically significant drop right there. Barley, not so much, and field pea, not at all. So really what this told us was that, okay, plasma is doing something, but we're not sure why or how. And I started wondering if some of the morphological features of the grain's surface might have to play a role in this. So again, I kind of went back to the basics where I started looking at a bunch of these grains under the scanning electron microscope. And the goal was to see if I could figure out where these pathogens were. And I started noticing a bunch of trends. Pathogens seem to be located in very specific areas of the grain. On Psyllium, the dorsal surface, as you can see at the top, is nice and smooth, but the ventral surface is this big cavity. There's this nice boat shape there. And it turned out 50% of the grains that I scanned actually had visible fungal infections in that cavity. So that's a nice little microbe hidey hole. Wheat had the same thing. Turns out that brush, the hairy bit on the end, it's chock-a-block full of microbes when you start to really look at it. And there's all kinds of other stuff in there. So there's all kinds of debris, bits of pollen, microbes. It is just super dirty. So I started wondering if that might play a role in what was going on. Different grains have different structures, which might be protecting some of the microbes. So I started out by kind of making up this assay. I had no idea if it was going to work, but it was really the only way I thought I could try to understand what was going on. So what I did was take grains and I popped them into well plates and completely encased them in media. And then every day for a series of days, I would go under the microscope and I would inspect every single one of these grains. Each one of those wells I divided into 4 quadrants, so I could look at them from one side, 2 faces, and the other side, 2 faces as well, and start to try to track where fungal hyphae were growing. Now, this took me weeks and weeks, but I finally got some data sets, and I was really excited when it came to maize, because if you did a time series of plasma treatment, you could see the visible number of hyphal colonies across all areas of the grain were significantly dropping down. Those are huge changes. And on maize, there didn't really seem to be too much of a relationship between the position of microbes on the grain. But maize is pretty nice and smooth all around, whereas wheat tells a very, very different story. We are not able to drop the concentration or the number of hyphal colonies down, but it does show that near the embryo, we have way less visible hyphal colonies then compared to different areas, especially the brush and the crease. So the brush, we saw a bunch of things growing in. The crease on the underside also tends to potentially be a nice hiding spot. And that led us to thinking, okay, well, we know there's microbes hiding out. We had a slightly difficult trying to culture them at the very beginning. What if we try an inoculation trial? What if we get rid of all of the pathogens existing on the seed and then we blast them with plasma and see what happens? So we decided we picked our 5 grain types and 7 economically relevant pathogens. 3 of those pathogens, the Aspergillus, the Penicillium, and the Fusarium, are also post-harvest problems. What they do is produce a bunch of different toxins that cause a lot of grain loss. We also chose things that were very relevant in South Australia because they can be passed on through seeds and also survive in crop residues. So, looking at that, we decided with some of these pathogens, particularly the Penicillium, we were going to put it through a time series of plasma treatments and see how that drops down. And sure enough, through 3 independent experiments and a lot of plates, we were able to show that yes, 90 minutes of plasma treatment drops down the concentration of colony forming units. It's significantly. We're talking drops from over 12 million down to like 8 or 10. That's absolutely huge. And then we also looked at Aspergillus on maize. And from that field P data with Penicillium, I kind of noticed that we were having big drops by 90 minutes, but what happens if I extended the plasma a little bit longer? And it turns out, sure enough, I was able to completely get rid of all microbes. All of the Aspergillus flavus was completely gone by 105 minutes. We're starting with concentrations of 3.7 million down to 0. So that was very encouraging that this technology might work. So we decided to put it up to a bunch of the big bad things. We have our Aspergillus that we redid on maize. We did bipolaris on barley and fusarium on wheat. And these are, with these fungi, we decided not to inoculate them with spores. Instead, what we did was grow hyphae over the tops of the grains, get the fungal colony to completely cover the grain. And that's when we used it with the plasma treatment. We also have Phoma and Penicillium. The Phoma was done in a liquid inoculum, and the Penicillium and the Aspergillus were done in airborne inoculums. If you look at what's happening, you can actually visually see some massive changes. On the Aspergillus on maize, you can see for untreated, which is across the whole top, we have spores all over the place. They look kind of like little raspberries, and they're nice and plump and happy. And then by 90 minutes of plasma treatment, they're still there, but they don't look so good. In fact, you can see they're starting to be shredded. They're shriveled up, and they're actually missing some of their surfaces. The same thing was present in psyllium inoculum with Penicillium. So the Penicillium spores, you'll see again, nice and plump and happy on the top. And then with plasma treatment, they're kind of like deflated little balloons. With Phoma on the field P, and I apologize because I put FOMO, not Phoma at the top. You can see on the untreated, again, you have these big plump happy spores, and with plasma treatment, it's almost like we've taken a lightsaber and chopped them completely in half. And finally, with the pathogens that we grew over the top of the grain, on untreated, you can see these gnarly infections that are just growing everywhere with spores in the middle of them on bipolaris. And then after plasma treatment, they're completely gone. We have shredded all of the hyphae and all of the spores. The same thing goes for Fusarium. Again, these gnarly infections on the surface, and in the right-hand side, you can actually start to see the hyphae penetrating the grain. But after plasma treatment, they're so shredded, they are not going to be viable from here on out. And with Fusarium, we actually got a little more excited because we realized that Fusarium makes 3 different types of spores. You have macroconidia and microconidia, and then you have chlamydospores. So the macro and microconidia tend to be the sexual reproduction phases of that microorganism, whereas chlamydospores are a vegetative spore, and this is the part of Fusarium that makes it very, very infectious. These are double-walled spores that sit down in crop residues and in soil and leaf litter, and they can actually survive for up to 20 years in the environment, waiting for the right conditions for an infection to take off. So we were able to induce the production of these on top of our grains, as you can see on untreated, and then after plasma treatment, we have shredded these double-walled, super persistent structures. So that made us pretty happy. And we decided, okay, well, if we can get rid of all of these pathogens in a synthetic system that we've created, does that mean we've now been able to figure out the right conditions to get rid of a huge number of pathogens? So we went to industry. We went to Viterra and we asked them to donate grain that was the dirtiest, grossest samples they could possibly find. So they were absolutely lovely and gave us a 20-kilo bag of some really gross grain. And it turns out we found all kinds of pathogens all over it. So we, through both plating and visual identification, as you can see some of the microscopy here, as well as ITS and NGS, in collaboration with some groups at Sardi, we were able to identify all of the different pathogens listed, which is a huge, huge mix. And we wanted to test the plasma system to see if we could clean this grain up and get rid of all of these things with plasma treatment, which was a much more complex system because not only do you have these pathogens interacting with the grain, but they're interacting with each other. So the first step was actually to compare the contaminated grain to food quality grain and see how many colony forming units, how many things we could get growing off the grain. And it turns out food quality, we're sitting around 950 or so colony forming units per mil, and the contaminated grain was pretty dirty because we're at 112,000. You can also see from the plates there, just putting the grain onto media, it just grew all kinds of grossness. So we put it through a series of plasma treatments. I extended the plasma out to 120 minutes, knowing how dirty and gross this grain was, to see if we could get rid of things. And sure enough, we had log 5 reductions. By 120 minutes, we got rid of anything that was a filamentous fungi. So I wasn't able to count colony forming units for bacteria or yeast because I couldn't really figure out what was going on. They kind of grew together and it was very hard to count. So the filamentous fungi, we annihilated by 20 minutes. You will see there's a bit of an anomaly at 90 minutes where the numbers do go up. And you can kind of see that's just based on 2 different counts. So there were 2 places with a few more spores. And that's really just a little bit of biological variation. Doing extra statistical analysis, t-tests and everything just shows that it's not statistically significant. But really what this is telling us is by 120 minutes, we're annihilating all of those pathogens. So then we took these data sets, I inspected the plates, and every single one of the 180 plates, I looked for and gave scores for all of those different pathogens that we were finding. And so we can look to see which pathogens were disappearing after different time points of plasma, which was very fascinating. By 90 minutes, we still had a little bit of stuff left. And by 120 minutes, we've gotten rid of most of it. So you'll see the Cryptococcus magnus is left, and that's an encapsulated super hardcore yeast. Again, one of those things I wasn't able to quantify with the colony forming units analysis. Bacillus safensis is a spore-forming bacteria that is so hardy NASA can't even kill it in its spacecraft clean rooms, which means Bacillus safensis has probably colonized both Mars and the moon. And we also had some mixed cultures of things that were still present. Aspergillus flavus, we had 2 plates with just a couple spores each, so we didn't completely get rid of it, but close enough. And we were pretty happy with these results. Just to back it up, just because we could, we did a little bit more microscopy to really show what was going on. And sure enough, the untreated samples were completely coated in microbes. And by 120 minutes of plasma, we've shredded them all. So really, what does this mean? Well, we've learned that The different structures and the morphology of the grain plays a role in where the pathogens may be and whether or not plasma can be infected. If there's hidey holes, well, it's going to be hard for plasma to reach them. You're going to need really long-term treatments, like 120 minutes for plasma to be effective. We also know there's a nice inverse relationship between plasma and pathogen viability. So the longer the plasma treatment, the fewer the pathogens. We have critically damaged and just annihilated some of these things, as we could see under microscopy. And we knocked out some of the worst post-harvest pathogens on real-life samples. The samples that Viterra gave us were things that they actually had to destroy. That was an economic loss for them. So we've shown that a real-world sample really can be turned around. Overall, looking at this entire study, we know that plasma treatment is definitely changing the physical properties of the seed. We're changing water sorption and the surface chemistry. We know that that might be impacting the growth and viability. So long-term plasma exposure is definitely not ideal for germination of domesticated grains, but maybe short-term worked. We saw in a few of the species that 15 minutes of plasma actually improved germination rates. It might still work for really difficult to germinate native species. These things sometimes have really hard surfaces that it's hard for water to penetrate. So it's definitely something to look into. We also know that plasma has been fantastic for surface disinfestation of all of these different microbes. We can get rid of most things. And it appears to be a great technology if you're looking at grain that's post-harvest and pre-food processing. So if we're going to be turning some of these things into flour, if we can plasma treat, not only are we bringing down the seed moisture content with the vacuum, but we are getting rid of all of the microbes. So that also starts to lead to a few future directions. If we're getting rid of those microbes and making these big chemical changes to the surface, can we get rid of the toxins those microbes can produce? A lot of those mycotoxins, potentially. Also, could we break down chemical residues on grain like glyphosate and the metabolite AMPA? Potentially as well. There's a lot of chemical reactions going on. Next, can the system be upscaled to handle much larger volumes? Can we move this very simple system, bring it to a level that it can actually be used in industry? And if we can't engineer it to be absolutely massive, could we look at it for developing countries, for subsistence farmers who are doing much smaller volumes of grain? And since this is only using 50 watts to generate the plasma, it's something that could be run off of solar panels. This is definitely a possibility. And then finally, can we use that plasma to break dormancy in Australian native seeds? Not really sure. These are all different directions we could take it in. But overall, this kind of sums up all of the research I've been doing, and all I can do is just say thank you to everyone on this campus who has helped me throughout this time. There are so many people that are not on this list, so I apologize if your name is missing, but I wouldn't have been able to do this without you. Thank you.
Perfect. Well, thank you so much. I think I'd like to see if there might be any questions. Let's see. We'll give you a bit of time to maybe submit any questions if you've got any. There we go. First one, could plasma treatment change ROS levels in the embryo?
Yes, I think it definitely could, and I think that might be one of the things that's going on. There's actually been a little bit of work done in a couple studies actually that came out this year within the last few months looking at ROS levels in the embryo. That could be one of the things that's working, but I think it's actually going to be a big combination of not only the ROS, but the way that water's entering. I think that's probably going to be one of the bigger things. Seedlings are actually pretty good at dealing with oxidative stress. They've been desiccated for so long that when they get hydrated, they kick into gear. There is a lot of changes in hormone signaling. And Phil, I've actually found a few more papers on that hormone signaling stuff. So yeah, if you want to chat about it, we definitely can.
Let's see. Could you increase the efficiency through evacuating the tumbler and introducing different gases, or by adding a small volume of hydrogen peroxide?
Yeah, so we actually did investigate using different gases in the plasma tumbler. So I tried using argon as well. And it turns out that air plasma, for some reason, is magical. It just works really, really well. And I think it has to do with the combination of reactive nitrogen and reactive oxygen species that we're creating from it. A water plasma, which has a lot more of the hydrogen, as well as obviously the reactive oxygen species that are being created, it wasn't really effective. Argon was really good at doing ion bombardment, but I didn't have as many chemical reactions, so the oxidation wasn't taking place. So we could try a few other gases. I haven't tried straight nitrogen yet, but it is a possibility. And we decided not to do, hydrogen peroxide was an option. That we decided not to actually look at it though, but because we're actually producing a crap ton of hydrogen peroxide in the plasma process anyway, that it really wouldn't be worthwhile. I'm already creating tons of hydroxide molecules and all kinds of other reactive things that are going on. But yeah, definitely something to think of. Thank you.
Let's see, then we've got one in the chat as well from Sarah Blake. Amazing research, Mel. What volume of seed were you plasma treating at any one time? And in terms of scale up, what volume of seed is currently possible to treat this way?
Sadly, right now we're on lab scale, so it's super teeny tiny. I'm working with only 100 grams of grain at once. There is another plasma tumbler that's actually located at UniSA, and that one's capable of holding about 4 kilos at a time. Unfortunately, my research for my entire PhD has been done in such a short time. If I had more time, I would have tried to get this instrument here and start doing multiple kilos at a time, but it just hasn't been possible. So I recommend anybody who's interested, go talk to Brian Code. He's looking for students.
So we've got Jacob Schultz. What is the scalability of the technology? Can you achieve this at an industrial level? But even then, does the low energy input translate when scaled? Do any large scale systems exist at the moment? I think most of that has been answered.
Yeah, there are, so people have been looking at this and using different plasma technologies. A lot of times people are using a different type of plasma generator, which is called a dielectric system. And so you have basically 2 planes, and what they're doing is putting seeds on conveyor belts and running them through a plasma plane like this, kind of going through, and they're looking at that on a mass scale. But it's been really, really difficult for them to get that to work. There has been a fair bit of research on it, which is actually why we started using a plasma tumbler, because it's a different system that's built into a drum essentially and constantly spinning the grain. So the plasma is interacting with all surfaces, not just 2 surfaces or even 1 surface. What we're thinking is, I guess, just imagining out there, yes, this technology is definitely scalable. There is use of massive plasma reactors. This type of radio frequency capacitive coupled plasma reactor has been used on very large scale. And I kind of envision a system that's essentially like a massive drum, kind of like what you get in a wine bladder press that kind of spins around, but it's kind of spinning around and also, I guess, having a field of plasma in there too.
Then Matt Solomon, do you think we can expect increased mutation from germinated seedlings due to genetic damage?
I don't think so. I mean, it's always a possibility, but I really don't think so, because what we're showing is that on the oxidative DNA damage, we weren't really getting that. Across the board, it was pretty inconclusive. A lot of the untreated grains had an equal amount of oxidative damage. So I would say we're probably not getting a lot of that done. And if it's a grain that's going straight into food production, I don't think it's going to be an issue no matter what. And then is there potential for controlling peanut allergies? I have no idea. I'm not really sure how peanut allergies work, so maybe.
Who knows? Who knows? Cool. Any more questions? Just give it another minute. I think that might be it.
Cool.
Let's see. Thank you very much. And you can find this webinar back later on as well, and hopefully the questions and everything as well. You can probably contact Mel if there's anything coming up later on.
Yeah, if you have any feedback, I'm submitting in 2 months, so I need to get feedback soon. Thank you.
Thank you so much, everyone.